Back. Today's we discuss about one of the ABCP transport CFTR. It is an exocrine tissue transport chloride and disease. Transport chloride and if it is mutant cause disease cystic fibrosis. It is mainly caused when the autosomal mutation on chromosome number 22. Okay, so let's get started here. If we talk about the mechanism of CFTR, it is 12 transmembrane oriented from one polypeptide. It means type 3 NNC both in the cytosol. If we talk about the Hardy-Wiener principle according, so Q square is equal to 1 upon 25. We know that the formula is that P plus Q is equal to 1. So Q is equal to 1 upon 50. So P is equal to 49 upon 50. So 2PQ is equal to 1 upon 25. So it is a Hardy-Wiener. It is autosomal. Okay. It is a 12 transmembrane. First 6 in T1, then cytosol A1, then 6 membrane T2, then cytosol A2. For activation of chloride and pump, first phosphorylation of regulation of domain serine residue. After firstly phosphorylate regulatory domain contain a serine residue. Firstly phosphorylated that serine residue. After phosphorylated, no channel open. It means not only serine phosphorylated is not requirement or not necessary to not is the main point to start the channel. Second, firstly phosphorylate the serine residue on the regulatory domain. After phosphorylated, then here 1 ATP used to phosphorylate serine, then 2 ATP come and bind nucleotide binding domain of A1 and A2. Cytosol site, after ATP bind in nucleotide binding domain, channel open, chloride transfer cytosol to lumen against the concentration gradient. Here cytosol to lumen transfer against the concentration gradient, delta G is positive. That why here you use ATP as an energy source. Mutation in the CFTR or CFTR defective gene include misfolded protein. If its A1 have a phenylalanine 508 are mutant, though chloride channel is mutant and called cystic fibrosis diseases. The misfolded protein deposit chloride and the cytosol here cytosol is already according to the normal way cytosolic concentration low in the chloride concentration low in the cytosol, but this is mutant, those chloride concentration high in the cytosol or reduced chloride and concentration in the lumen or digestive tract or air base of lungs. So if mutants of chloride and not transfer of water, not so mucus is thick, so it is a favorable to grow colonies of bacteria. Reduce fluid of tract, resulting mucus become dry, and bacteria which are responsible for the diseases are which are grow in their environment: Pseudomonas aeruginosus and Streptococcus aureus area, not inhibited by and these are affected respiratory tract and they are responsible for the respiratory tract failure. Here how the channel is work. Here is the 6 T1, then A1 cytosol. If mutation in A1 phenylalanine 508 residue, then the mutation is caused. Then T2 in the membrane and A2 in the cytosol. Here R domain, it means regulatory domain serine residue. Firstly it phosphorylated, then secondly for the activation attach ATP on nucleotide binding domain. Okay, here mention the nucleotide binding domain. 2 ATP bind on the two different sides of the nucleotide binding domain, channel open against the concentrations chloride and move between cytosol to lumen. Transport across the epithelium, absorption of glucose from food to blood. Here is the only is the process and how the chloride and shift mutation. If the mutation the phenylalanine 508 and another is the glycine 551 is also mutated, so cause CFTR cystic fibrosis. Okay, transport, if we talk about transport across of epithelia, absorption of glucose from food to blood, fluid, internal microarrays, sodium glucose transport, it means import SGLT1 and 2, and 2 sodium and along 1 glucose against the concentration, it means sodium and along and 1 glucose against the concentration inside the cells, sodium and inside. So here sodium potassium pump, sodium outside against the concentration, movement into out against, and potassium movement against the concentration out to in. So here ATP used in both for the sodium ion movement and potassium ion movement. Okay, here's the way plasma membrane, lumen, blood. Now glucose come inside against the concentration of glucose high in the cell. It means the concentration is reached on 3000. So GLUT2 active, where GLUT2 km value is 17 when glucose high in cell, because it came 17. So when the glucose concentration is high, then GLUT2 active in the liver, and it take the glucose from the blood in liver and store in the adipose tissues. Glucose sodium ions import 2 sodium ion along and 1 glucose against cytosol by symporter SGLT1 and SGLT2. When glucose high in the cell, GLUT2 and 2 sodium ion high in cells, sodium ion high inside. So maintain concentration sodium and require sent outside. Run homeostasis sodium and go outside against the concentration by use potassium ion in the out against the concentration by sodium potassium. ATP is require sodium and low in the cytosol, potassium and high in the cytosol, sodium and high in the blood and potassium and low in the blood. High glucose when eat food. How this the ATPase sodium potassium pump sodium potassium ATPase work? Here it is the cytosol. We know that the sodium ion concentration low in the cytosol, potassium ion concentration high in the cytosol. Blood have a high sodium ion concentration and the potassium ion low concentration in blood. And the lumen of intestine, here 2 sodium ion and 1 glucose. 2 sodium ion along the concentration, 1 glucose is against the concentration. And here is the blood movement of the sodium in between sodium potassium by sodium potassium ATP. Sodium and potassium and move both are against the concentration. Use ATP as an energy source. Sodium and against the concentration cytosol to blood, and potassium and against the concentration blood to cytosol. Here titration sodium and low in cytosol, potassium and high in the cytosol. Blood high sodium, low calcium and potassium, intestinal lumen sodium high, dietary glucose high, and sodium and chloride concentration high. Now we talk about how the acidification of the stomach lumen done by chief cell and parietal cell stomach pH lumen 1 to 1.5, which is a maintain potassium ion ATPase or hydrogen ion. It means hydropotassium H and potassium and ATPase के through ये maintain होगा. Now here blood CO2 partial cell transfer and partial parietal cell by diffusion. Here water and carbon dioxide react and it convert into the bicarbonate and HN. Here HN react SCO3 outside in the blood and it move in the blood, and the chloride and inside the parietal cell, and his CFTR run. That's why chloride and come into the lumen from the cytosol against the concentration by the CFTR, and HN sent outside, and potassium ion come against the concentration from outside to inside. Here 4 to 139 against the HN chloride and HN and chloride ion come outside and form HCl in the lumen of stomach, and HCl is the main HCl pH is more acidic, not a slight it's a tongue twist. It's HCl is a highly acidic. HCl and HN come from inside to outside, and potassium ion come from outside to inside. Both aqueous concentration use ATP because both move against the concentration, and HN potassium ion ATPase p-type inhibitors. It means it work like a p-type inhibitor, so it inhibit by inhibit of HN and potassium and ATPase are called proton pump inhibitor. So blood side chloride and bicarbonate anion exchange, lumen chloride and channel or potassium HN ATPase. Here this work, how this work. Now we talk about the last topic, aquaporin, water transporting channel. It also move water, glycerol, urea, arsenate across the cell via apical plasma membrane, basolateral membrane, apical membrane or plasma membrane. Aquaporin is a tetramer made up monomer is a 4 large helix and 2 small helix. It means it contains 6 helix. Have a conserved sequence NPA. NPA represent here N for asparagine, P for proline, and A for alanine. And the two another amino acid R and H. It means R represent arginine and H represent histidine, located in the cavity. Major regulate cavity NPA because and the asparagine react hydrogen bonding with water, and proline help in the curving, and alanine is small due to the alanine, it's a small side chain, that why alanine fit in the curving nature. Okay, now water follow maintained by proton hopping, it attach water molecule. So arginine is prevent the proton hopping because cell is a very delicate. If continue water flow, so it rupture the cell. To arginine have to prevent the water hopping, that's why the water come in cell very smoothly from ancha, so it reach and asparagine. Here the amino acid which I used, they coded their number: histidine number 180, asparagine 192, arginine 195, and aspartate 76. Okay. Now it's a side topic, the potassium ion channel. It is also a part of the aquaporin. Here's the 12 transmembrane 662 A site 2 nucleotide. It is the CFTR. Here's the channel. If channel is the ligand respond to ligand, and the channel to membrane voltage gate ion channel. If GABA, acetyl-CoA, DOPA, and glycine. Channel open with response to ligand is called ligand bind channel. If they are the voltage sensitive, the voltage gate, and if they are gated sensitive, the gated. Sodium and voltage gated ion channel found in skeletal muscle, neuron, cardiac muscle. Potassium and voltage gated channel found in neuron. Okay, remember that sodium and voltage gate found in the skeletal muscle, neuron, and cardiac muscle, and potassium and rated voltage channel found in the neuron. GABA, glycine, serotonin are the receptor of the ACH. GABA and glycine are anion specific, and acetyl-CoA and serotonin are the cation specific. Cyclic GMP, cyclic MP, ATP, IP3 also act the ligand for gated ion channel. If we talk about the toxins, the toxin inhibit transport. If we talk about the tetrodotoxin, it contained from the pufferfish. That's why pufferfish. The last century there are the many events when when the public is not the pufferfish contain a tetrodotoxin, they eat the pufferfish without it untoxified. That's why they kill many animals, they kill many humans being, and the wastage also eat by the roadside animals, so they also die. So now after they identify the pufferfish contain the tetrodotoxin, so for the cutting of the pufferfish, the chef is qualified to how to detoxify the pufferfish. Okay, so tetrodotoxin is a pufferfish, cause inhibition of sodium and potassium ion channel, and antitoxin marine plankton inhibit neurotransmitter muscle contraction. Dendrotoxin is contained from the black mamba. Snack toxin is inhibit voltage gated potassium ion channel that are found in the neurons, and the cobra toxin, bungarotoxin block the ACH receptor and bind with the great affinity with the acetylcholine. Okay, if you want to the purify acetylcholine, so use bungarotoxin. Okay, the extreme highly affinity of bungarotoxin of acetyl receptor is KD is a 10 to the minus 15 mole, has proved useful experiment. The radio labeled toxicity was to across the receptor due to the purification. It is done. Then let's discuss something about here is some part of the ATP binding cascade channel. So let's discuss it also. So here ATP binding cascade transmit the transfer hydrophobic molecule and chloride and out of the cell. If we talk about the ABCB1, it's also known as the MDR1. It means multiple drug resistant. Most of the drug are antibiotic hydrophobic, or that's MDR1 sent outside. So antibiotic not effect due to the MDR1 because it expose lipophilic drug. Okay, we talk about the MDR2 in liver. It expose phosphor. It means MDR1 expose lipophilic drug and MDR2 expose phosphor. It attach